Uh, it's the 5th of July 2020. Independence Day is approaching very rapidly, and that, as you all know, is the 15th of August. But what you do not know, and I can bet on that, is as to why that particular day, the 15th of August, was chosen to be our day of independence. Well, the choice was not made by Gandhiji. It was not the decision of our first Prime Minister, Jawaharlal Nehru, nor was it the choice of Mr. Jinnah Sahab. The decision for the day was made entirely by one man, and he was then the Viceroy of India, Lord Louis Mountbatten. Mountbatten probably came to India on the 22nd of March 1947. Before he came, he had, as he said, put in a rider to the Prime Minister, Clem Attlee, that he would agree to be the Viceroy of India, provided every decision of his was not vetted by His Majesty's government, which meant that he would be in a position to take decisions on his own without interference from the Prime Minister or his cabinet. So why did Mountbatten choose the 15th of August? The answer is simple. The 15th of August is the day the Japanese formally surrendered to the Allied forces. And that was the end of the Second World War. It was Emperor Hirohito who eventually realized that Japan was on its knees. Two bombs had been exploded. Both were atom bombs. The first was on the 6th of August, 1945, I think at 8:15 a.m. The second was in Hiroshima on the 9th of August. And by the 15th of August, Japan had capitulated completely. Formal surrender was done on the 2nd of September on a ship called the Missouri. But the Japanese surrendered to the Southeast Asian forces on the 2nd of September 1945 in Singapore. The instrument of surrender was signed both by of course, by the Japanese and presented to the Supreme Commander of the Southeast Asian Forces, Lord Louis Mountbatten. So for Mountbatten, the 15th of August was probably the highest point of his life. And therefore, we got our independence on the day the Japanese eventually surrendered to the Allied forces, bringing to end the Second World War. Now, I mentioned the 15th of August because a very interesting letter has been dispatched on the 2nd of July this year by the Ministry of Health and signed by the Director General of the Indian Council of Medical Research, that is the ICMR. The letter is astounding. It is written to dear colleagues. Please, I emphasize the word dear. The letter is addressed to dear colleagues, and it says that it is envisaged that a vaccine would be used publicly not later than the 15th of August 2020, after all clinical trials are completed. So the Health Ministry is imagining that a vaccine will be ready for public use in 5 weeks from now, after all clinical trials have been completed. Now you must keep in mind that we have not had a single clinical trial of any vaccine in India as of now. The company which is partnering with the ICMR is the Bharat Biotech International Limited, and the CMD has given an interview yesterday to the Financial Express in which he has clearly stated that the vaccine will be ready by next year. which is, of course, a very realistic presumption. Now, back to the letter. The letter also writes in its last paragraph that kindly note, non-compliance will be viewed very seriously. It's almost a threat, because what the author of the letter is demanding is that there should be, in fact, he uses the phrase, you are seriously advised or strictly advised to fast forward all regulations and protocols to begin the clinical trial. Now, we all know that we have a pandemic on our hands. There is a major problem. There are serious problems because of this pandemic. But the fact that the DG of the ICMR is writing such a, frankly, absurd letter, wherein he's saying that he wants a vaccine for public usage before the 15th of August this year, it's not 2021, but 2020, is a bizarre proposition. Now, I must walk you through what or how a vaccine is developed. You see, the first thing is a vaccine is made wherever it is in a laboratory. Then the vaccine is tested on animals, like the Oxford University has done. Not only has it tested the virus on monkeys, it also has published a preprint. The preprint says that the Oxford University researchers have tested 9 monkeys, 6 were inoculated, or rather given the vaccine. 3 monkeys served as controls. Then all 9 monkeys were inoculated by the SARS-CoV-2 virus. And after a span of 1 month, it was found that all the monkeys, that is the monkeys given the vaccine and the monkeys not given the vaccine, all of them developed infection. So now that was a downer. But the saving grace was that when the monkeys were euthanized, or rather sacrificed, they found that of the 3 control monkeys, 2 had pneumonia, whereas all the 6 monkeys who had been vaccinated, none of them developed pneumonia. So the spin has been by the Oxford University researchers that, look, the vaccine will not prevent infection, but it will definitely diminish the symptoms. It may not help completely, but it certainly will, or possibly will, prevent pneumonia and other serious complications of the SARS-CoV-2 virus. The important thing to remember is that the experiments have been done on monkeys by the Oxford group, which has, I think, teamed up with AstraZeneca, and they will begin their human trials next month, that is in August. Probably the phase 1 trials are going on. So let me elaborate more on the stages of testing a vaccine. After you have found the vaccine is effective, or rather is capable of producing antibodies in an animal, the animal could be a rat, it could be mice, it could be a guinea pig, a rabbit, or a monkey. Once you have found that the vaccine is capable of producing adequate antibodies and also is safe in the animals, you move on to the phase 1 trial. The phase 1 trial picks up or takes about 45 or 60 participants. They all should be healthy, and above all, they should not be infected by COVID-19. They are given the vaccination, and then you wait and watch for a month for adverse effects. Now here I must point out, the Chinese have already published a paper in the Lancet, way back, I think on May 22nd. I will provide the link of that study. This was published in the Lancet, so kudos to them. The study is on 106 participants who were given 3 doses of a vaccine. I will talk about that particular vaccine later. The 3 doses was a small dose, a medium-sized dose, and a large dose, and they found that 80% of the participants developed some adverse effects. Remember, this is a phase 1 trial, but the side effects were mild, consisting of pain at the site of injection, or systemic features like fever, headache, body aches, and myalgia, but nothing beyond that. There was no serious side effect. The mean age of the participants was 37 years. Half of them were males. So the take-home message or the conclusion by the researchers was that their vaccine is safe in human beings. So this probably was the first human trial on a COVID-19 vaccine, which has been published way back in May 22nd in The Lancet. Now, the vaccine which was used by the Chinese in this study was a vector-based vaccine, which means what they do is they take a gene of the COVID-19, a bit of that gene, and put it inside an adenovirus. The adenovirus is the common cold virus, and this virus is taken from a chimpanzee and then made capable of infecting human beings, and this is a harmless virus. So the harmless virus, the harmless adenovirus, like an airline or like a automobile, carries a passenger. The passenger is a gene of the COVID-19 virus, a bit of that gene, and this bit of gene is capable of giving directions to make the spike portion of SARS-CoV-2. Now, as I have told you earlier, the spike portion is the one which is capable of eliciting an antibody response, and also this is the bit which allows the SARS-CoV-2 virus to penetrate human cells. So the vector-borne virus, or rather the virus which has been used by the Chinese, and in fact I think the same technology is being used by the Oxford University group. A virus is injected, that is the adenovirus. This virus carries a bit of the gene of the SARS-CoV-2 virus, and that bit of gene then gives direction to the human cell to produce the spike portion of the COVID-19 virus. And now we have antibodies produced, and these antibodies in the human body will prevent infection in the coming months by COVID-19. Now, the important thing to keep in mind is that the Chinese study also states that all the participants developed adequate antibodies, not only the B variety, that is the IgG and the IgM antibodies, but also the T cells, which was specific for the SARS-CoV-2 virus. Now, the Oxford group will begin its trials next month, as I've said. I understand this month Pfizer begins its human trials, and in September we will have Johnson & Johnson beginning human trials. There are more than 150 vaccines right now which are being developed for this pandemic, and by and large a lot of them have funds by the governments of the United Kingdom, by the American government, the NIH, the CDC, and of course the WHO, as also the Gates Foundation. So the point to remember is that first you have the animal studies, then you have the phase 1 trial in which you check for the adverse effects of the virus, of the vaccine to prevent the virus in about, let's say, 50 people. Next you move on to the phase 2 trial. The phase 2 trial checks for efficacy, that is whether the vaccine works or not. Now this time you take a larger group of participants, maybe 500 or maybe less or maybe more, but that number should be adequate. Now here you inject the vaccine, maybe depending upon the technology, some would need 1 dosage, 1 injection, and the other technology may need 2 injections, the second one a fortnight later. Once the vaccine has been given, now you wait and watch for the efficacy, which means whether the vaccine actually prevents future infection by the virus. So again, the participants must be healthy, they should be seronegative, which means that they have not been infected by COVID-19. Now, this is a problem actually to pick up so many people for the phase 2 trial. But of course, it has to be done. And you have to wait for at least 8 weeks or 10 weeks or 12 weeks, maybe 4 months to find out, or maybe even more, maybe 6 months. Logically, you will need at least 6 months to find out whether the vaccine works, because you have to wait. See, what you're doing is you are injecting normal human beings with a vaccine. Now you have to wait whether the vaccine actually prevents infection or not in these people. Then you come to what is called the third phase. The third phase is, provided you find that the vaccine has worked in human beings, you get more people. Now, the phase 3 trials, you need possibly 20 to 30,000 participants. Again, all healthy, all should be seronegative, all should not have been infected by COVID-19 in the past. All right? So, supposing you pick up 30,000 participants, in 20,000 you will inject the vaccine, and in 10,000 you will inject a dummy. So this will become a placebo-controlled trial, a randomized study. And in case even the injector does not know what he's injecting, then it will become double-blind, which means neither does the participant know, nor the person giving the injection knows what he is injecting. So that is a double-blind, placebo-controlled trial. And I think these trials are what I'm talking about, that AstraZeneca will begin next month, and Johnson & Johnson in the month of September, and Pfizer will start in July itself. Now you can understand that when you inject the vaccine in a normal participant, right, and then compare it with a dummy injection in another normal participant, you have to wait for some time to see whether the vaccine actually prevents infection. You see, this has to take 6 months or 4 months or 3 months. You have to wait for 4, 5, 6 months. Logically, that's common sense. You don't say that, okay, I've injected today on the 1st of August, and by the 7th of August, I've made a decision that the vaccine works. For the vaccine to work, there has to be a significant difference in the development of the disease between the control group, which has been given the dummy vaccine, as opposed to the group which has been given the vaccine. So that will take time. That is the reason why all serious scientists, all serious investigators have been telling the public again and again that please wait for at least 6 months to a year for a proper vaccine to be developed. Because if you shortcut, despite the fact that there is a pandemic, despite the fact there is an uproar for some kind of a vaccination program to begin, we cannot take shortcuts. Vaccines have adverse effects, and these adverse effects do not develop immediately. You may develop some pain in your shoulder or in your thigh where the vaccine is given, but there can be other serious adverse effects which have to be looked into, which have to be kept under wraps, and these could be a chronic autoimmune condition or even a cancer. Now, I'm not an anti-vaxxer. I am absolutely not an anti-vaxxer, but it is absolutely essential to explain to you that here, corners cannot be cut. We cannot take shortcuts to develop a vaccine, because we are, let me use the word, envisaging that the vaccine will be administered to just about everyone on the planet. So we're talking about 6 billion injections. The question of developing so many vaccines, mass production, is a different subject. So what I am emphasizing is right now that vaccines are being developed. Some companies are on the brink of beginning human trials, but this particular letter, which states that the vaccine would be launched publicly in this country within 5 weeks, after completion of all clinical trials, is something truly amazing and something which was quite unnecessary. And this is addressed to the dear colleagues and marked to the CMD of the company, that is the Bharat Biotechnical. I think his name is Dr. Krishna Ella. And as I've said just now, Dr. Krishna Ella himself has said yesterday in an interview published by the Financial Express that he expects he expects that the vaccine would be ready by next year. And that is a conservative, a very conservative estimate. And also makes some sense. The CMD is clear about what he is talking about. The vaccine which will be used by this company is the inactivated vaccine. So let me talk about the different kinds of vaccines. I will make it very simple for you. The regular vaccines which you've heard of for measles, mumps, rubella, these are inactivated vaccines. That means the bug, the germ or the bacteria is inactivated, is killed by some chemical, right? There's always a danger that some would not be killed. So therefore, that disease can erupt. But however, this is the technology which will be used by the Indians. They have got the strain from the Pune Institute of Virology. They have the SARS-CoV-2 gene strain. They have the gene. They have the material. They have been able to inactivate it. And as per press reports, they have tested them on some mice and rats and guinea pigs. Of course, there is no paper published. These are all press reports, right? So you have the inactive vaccines. Then you have what are called the live attenuated vaccines, which means the germ is alive but has been weakened again by some technology, and that is injected. The third kind, and which is the most exotic, which has never been used before, that is the messenger RNA technique. Here, the messenger RNA itself of the virus, that is of COVID-19, is injected into the human body. That messenger RNA then directs the human cell to manufacture the spike component of SARS-CoV-2 or the COVID-19 virus, and thereby antibodies are produced. And the fourth kind of technology is what is being used or will be used is actually being used by the Oxford group, and the Chinese have also published a paper that is the vector-borne. That means the virus is carried in another common cold adenovirus, which is harmless. But the problem is that it will carry the gene to make the spike, but it cannot be re-injected. So therefore, you cannot give a booster dose because once you've injected this particular adenovirus in the human body will become immune to the adenovirus also. It definitely will produce antibodies. So therefore, we have inactivated vaccines. We have live attenuated vaccines. We have the messenger RNA vaccines, which are also under development. I understand that there's a snag, and that this particular technology is being used by the Moderna company. And the fourth is the one I've just mentioned, that the virus is carried by another virus. A part of the gene of the COVID-19 is carried by a common flu virus that goes into the body. The gene is released, and that gene then ensures that the body produces the spike portion of the SARS-CoV-2 virus, which then produces the antibodies. So we await these results with bated breath. We are all very anxious that an effective vaccine emerges on the landscape as quickly as possible. But we must ensure as a community, as people, that we don't take drastic shortcuts. We don't cut too many corners and land up with a vaccine which probably is partially effective, or worse, has serious adverse effects. I finish this talk by reminding you that even today, after more than 30 years, more than 35 years, there is no effective vaccine for HIV. There is no effective vaccine for malaria. In fact, there is no effective vaccine for dengue fever also. All right? So thank you very much for listening to me.